I am Dr. Khokon Kumar Dutta. I have a Christian name, Dr. David Boone. I am currently working as assistant professor in the Department of Biochemistry and Molecular Biology in Bangabandhu Sheikh Mujibur Rahman Science and Technology University, Gopalganj, Bangladesh. The title of my discussion is Role of MicroRNA-34c in reproductive successes and embryogenesis, at least from one cell zygotic state to implantable embryo formation. Khokon Kumar Dutta et al. Published an article in 2007 in the Journal of Cancer Science entitled Association of MicroRNA-34a overexpression with proliferation is cell type dependent. We for the first time experimentally confirmed the presence and expression of MicroRNA-34c in different rat organs. Internationally, this was the very first example of experimental validation of expression of MicroRNA-34c in any living organism or in any in vitro system. The experiment was inspired by the existence of MicroRNA-34c in the result of the gene prediction algorithm and its predicted location in the genomic map. The predicted secondary stem loop structure of MicroRNA-34c was also available in Sanger database of microRNAs. Globally, we for the first time succeeded to detect the presence of MicroRNA-34c in the testis. Testis showed the unusually high level of expression of MicroRNA-34c in comparison to any other part of the body or in comparison to the expression of any other member of the MicroRNA-34 family in the whole body of rats. In our experiments, we could also detect the expression of MicroRNA-34a and MicroRNA-34b in the testes. This was also the first time detection of MicroRNA-34a and MicroRNA-34b in the testes internationally. We also detected the expression of MicroRNA-34a, MicroRNA-34b and MicroRNA-34c in the lung tissues of rats. We also succeeded to find out the critical involvement of MicroRNA-34a and MicroRNA-34c in cancers. Internationally, we also for the first time tried and succeeded to relate embryonic stem cells and cancer cells in terms of the expression profiles of the members of MicroRNA-34 family. In 2020, it is almost clear that MicroRNA-34 plays key roles in cancer stem cells of almost all types of human cancers, irrespective of the cell, tissue, and organ types of origin. Wei-Min Liu et al. Published an article in January 2012 in the Journal of the Proceedings of the National Academy of Science of the United States of America, PNAS, entitled Sperm-borne MicroRNA-34c is required for the first cleavage division in mouse. The group detected the high level presence of both precursor and mature microRNA-34c in the one-cell stage of zygote. They also detected the high level of both precursor and mature microRNA-34c even after inhibiting the production and presence of microRNA-34c in the oocyte or egg. So they confirmed the paternal or male sperm as the sources of both precursor and mature microRNA-34c in the one-cell zygote. They also detected the presence of high level of microRNA-34c in the oocyte or egg-bound sperm during the event of fertilization. The depletion or inhibition of microRNA-34c in the one-cell zygote significantly reduced the cleavage into two-cell embryo. They also found that the absence of microRNA-34c inhibits the genome or DNA synthesis and replication needed during the division from one-cell zygote into two-cell embryo. They also reported in their article that microRNA-34c exert its effects partially by interacting with B-cell leukemia or lymphoma 2, BCL2. They also noted that there may exist other partners of microRNA-34c in the zygotic state other than BCL2, like MYC and others. Allison Tscherner et al. Published an article in September 2014 in the Journal of the Reproductive Biology and Endocrinology entitled MicroRNA-34 Family Expression in Bovine Gametes and Pre-Implantation Embryos. The group reported critical roles of microRNA-34A and microRNA-34c in both male and female gametogenesis and pre-implantation embryos. They reported the variation in patterns of expression and involvement of microRNA-34A and microRNA-34c between bovine and mice. They also reported the high abundance of microRNA-34c in the bull sperm in compared to other microRNAs and the critical roles of microRNA-34c in the pre-implantation embryos of bovine. Successful fertilization is a very important tool for livestock. At present, qualitative inspections of sperms like morphology, motility, and concentration of sperms are made before selecting for fertilization. These parameters does not always exclude the failure in successful fertilization and subsequent embryonic development. The group suggested to use the level of microRNA-34c in the sperm as the critical parameters of fertility in addition to the currently used parameters. Senlin Shi et al. Published an article in June 2020 in the Journal of Life Sciences entitled The Effect of Sperm microRNA-34c on Human Embryonic Development Kinetics and Clinical Outcomes. The group investigated the important roles of microRNA-34c on the success of in vitro fertilization IVF. They used time-lapse imaging technology TLI to monitor the embryonic development. They studied 58 IVF couples. They found that the high presence of microRNA-34c in the sperm was positively correlated with a high quality blastocyst formation and a successful pregnancy rate. The microRNA-34c level in the male sperm and in the zygote was negatively correlated with the developmental kinetics of human embryos. They suggested T5 as the critical point to determine the success of fertilization and pregnancy. It is to mention here that the morphology of the embryo and the time required for the blastocyst formation is the critical determinants of the success of fertilization and pregnancy. Both the cleavage rate and the high quality embryo are lower in the MicroRNA-34c siRNA treated group or in the MicroRNA-34c function depleted group. They suggested that the level of MicroRNA-34c in the human spermatozoa can be used as an indicator of IVF outcomes. Bo Wang et al. Published an article in 2014 in the Journal of Cellular Reprogramming entitled MicroRNA-34c Expression in Donor Cells Influences the Early Development of Somatic Cell Nuclear Transfer Bovine Embryos. One nucleus from diploid somatic cells transferred to the enucleated egg, a state like fertilized egg or zygote can be created using some voice-like electrical pulses to push in the direction of embryonic development to create the whole body of an organism. The scientific community of the world are well informed about it, as the famous cloned animal sheep Dolly was created using this type of procedure and principles. The group found that doxycycline, DOX, induced MicroRNA-34c expression in the donor cell enhanced the developmental potential of the somatic cell nuclear transfer SCNT embryo. Like the high level of presence of MicroRNA-34c in the sperm, high level of presence of MicroRNA-34c is needed in the donor cells for somatic cell nuclear transfer and subsequent successful pregnancy. They reported that high presence of MicroRNA-34c in the donor cell affect the epigenetic status, especially the histone methylation and the apoptosis in the course of the development of the somatic cell nuclear transfer embryo. Xavier Munoz et al. Published an article in December 2015 in the Journal of Nature entitled Altered MicroRNA Signature of Developing Germ-cells in Infertile Patients Relates to the Severity of Spermatogenic Period and Persist in Spermatozoa. The group identified MicroRNA-34c among the few microRNAs that account largely the spermatogenic period in the infertile men. They described a panel of the expression level of three microRNAs, microRNA 449A, microRNA 34C5P, and microRNA 122 as the predictive test for the presence of mature germ cells in the testis or testicular biopsy. They discussed that the roles of MicroRNA-34c is critical for the differentiation and survival during spermatogenesis. Ashok Agarwal et al. Published an article in 2015 in the Journal of Reproductive Biology and Endocrinology entitled A Unique View on Male Infertility Around the Globe. They described the global picture of male infertility. Infertility affects an estimated 15% of couples globally, amounting to 48.5 million couples. Males are found to be solely responsible for 20 to 30% of infertility cases and contribute to 50% of cases overall. However, this number does not accurately represent all regions of the world. So the detailed exploration of the roles of microRNA-34c in reproductive biology may help to overcome this big problem of the humanity. Finally, I would like to wish happy and prosperous days for myself and you everybody. Thank you.